In Alzheimer's disease research, two massive fields of study operate in complete isolation, despite the fact that the biological structures they study are physically touching. The first field focuses on the innate immune system. Here, the primary actor is the microglion, and specifically a molecular sensor inside it called the NLRP3 inflammasome, which assembles into a structure known as the ASC Speck to drive inflammation. The second field focuses entirely on the extracellular matrix. Their subject is the perineuronal net, a highly specialized condensed lattice of proteins and sugars that wraps and protects fast-spiking parvalbumin interneurons. Researchers studying this matrix have proven that during the early onset of Alzheimer's, microglia physically strip this protective net away from the neurons. To understand how the disease actually takes hold, we have to bridge the molecular gap between these two isolated structures, between what the immune system creates and what the brain's matrix loses. The current literature tries to bridge this gap by stating that an inflammasome-primed microglion shifts its output to degrade the net. That explanation relies on microglial activation, a placeholder term. It describes a biological state, but it does not describe a specific druggable pathway. Matrix researchers have successfully identified the exact downstream effectors, specific proteases known as 8-MTS and matrix metalloproteinases, or MMPs, that physically cut the net. But they do not know the transcriptional switch inside the microglion that actually commands it to secrete those specific proteases. Inflammasome researchers, meanwhile, know exactly how the immune switch works, but they track its output only as far as the release of inflammatory cytokines, ignoring the extracellular matrix entirely. Traced completely to its molecular source, the inflammasome emerges as the specific, powerful engine of perineuronal net degradation. If these two frameworks are coupled, the loss of the net and the activation of the immune system become a single pathological event, viewed from two different ends. We can trace this connection by mapping the forward arc of the mechanism. Inside the microglion, the NLRP3 inflammasome assembles and activates an effector called caspase-1. Caspase-1 then matures and releases the cytokine interleukin-1 beta. To see what interleukin-1 beta does next, we borrow directly from the most thoroughly characterized matrix pathology in biology, articular cartilage destruction in osteoarthritis. In that system, interleukin-1 beta binds to its receptor and triggers a signaling cascade to the nucleus. That cascade induces a highly specific secretory output. The microglion begins pumping out ATAPs4, ATAPs5, and MMPs into the synaptic space. Those enzymes target the perineuronal net. The ATAPs proteases specifically locate and sever the interglobular domain of aggrecan, the primary structural core protein of the net. Because this targeted enzymatic cut severs the structural core but leaves the outer glycan coat temporarily intact, early damage to the net is frequently missed by traditional tissue stains that only bind to the sugars. Interleukin-1 beta is the master transcriptional inducer that explicitly arms the microglion to digest the net. This connection tightens when we examine how the inflammasome cascade is triggered in the first place, specifically through lysosomal rupture. When a lysosome ruptures inside the microglion, it releases an enzyme called cathepsin B into the cytosol. There, cathepsin B acts as an intracellular danger signal, triggering the assembly of the NLRP3 inflammasome. But cathepsin B is also secreted directly into the extracellular space, where it functions as a potent protease capable of digesting the proteoglycan core of the net. The exact same enzyme acts as the alarm for the inflammasome inside the cell and the executioner of the matrix outside the cell. This brings us to the return arc. We have to account for what happens to the physical pieces of the destroyed net. Under normal conditions, the intact, high molecular weight hyaluronan backbone of a healthy net is immunologically silent. But when 8-MTS and MMPs destroy that structure, they generate a specific fragmentome. The net breaks down into low molecular weight hyaluronan oligomers, lectican fragments, and a glycoprotein called tenascin C. In peripheral sterile immunology, we know exactly how the immune system responds to these specific pieces. They act as damage-associated molecular patterns. They bind directly to toll-like receptors on the microglion and interact with the NLRP3 sensor itself. The return arc also involves a severe biological cost to the parvalbumin interneuron that the net used to protect. The intact net functions as a critical antioxidant shield. Stripping it away exposes the fast-spiking neuron to immense oxidative stress. As the unshielded neuron struggles, it leaks mitochondrial reactive oxygen species and ATP. Both of these molecules are established potent triggers that fire the microglion inflammasome all over again. The fragments generated during the net's destruction act as the precise ligands required to prime and fire the microglion again. When we view the forward and return arcs simultaneously, a clear mechanical circuit emerges. This inner loop operates sequentially. Matrix fragments fire the inflammasome commands the proteases, and the proteases generate more matrix fragments. This creates a tight, highly localized circuit, running underneath the much larger, slower feedback loop where amyloid beta diffuses out to the excitatory pyramidal cortex. In biological circuits, a loop with a shorter path length possesses a higher gain and a lower activation threshold. This closed circuit explains the clinical irreversibility of the disease. Past a specific tipping point, removing the upstream amyloid trigger will not stop a loop that now feeds itself entirely from the dying matrix. This inflammasome matrix loop offers a mechanism for the anomaly of cognitive resilience. We know certain human brains carry massive amyloid burdens, yet they maintain pristine perineuronal nets and normal cognition. The matrix proteases, ADAM-TS and MMPs, are not inherently pathological. The brain strictly requires them for normal physiological matrix turnover and neuroplasticity. If the exact same enzymes perform both functions, we have to identify what separates healthy matrix remodeling from pathological stripping. This split-screen visualization tracks what we call inflammasome tone. The difference between remodeling and stripping is determined by the amplitude of the interleukin-1 beta signal. Keep the tone low, and turnover remains controlled. Push the signal over the threshold, and you trigger runaway catabolism. Cognitive resilience occurs in a brain where the innate immune signal is constrained below that runaway catabolic threshold. Resilience means the matrix never enters the feedback loop. Because this circuit is a proposed model synthesized from two separate fields, this matrix table grades the evidence for each arc. The forward arc is textbook biology in osteoarthritis, and the return arc is proven in peripheral immunology. However, both remain inferential extrapolations until they are fully mapped in vivo within the Alzheimer's brain. We also have to account for the parallel effects alternative. It is possible that amyloid independently activates the inflammasome and independently drives net loss, without the two forming a closed local loop. But even if the closed loop fails, this synthesis achieves something crucial. It provides a specific, druggable molecular identity to replace the vague concept of microglial activation. This leads to an ultimate falsifiable prediction that unites both fields of research. If we administer selective inflammasome inhibitors during the latent interval, before the parvalbumin neurons die, we should observe a halt in aggrecanase activity and the preservation of the net. This approach effectively turns existing anti-amyloid inflammasome drugs into novel perineuronal net-preserving therapies. The net is stripped by an enzyme, the enzyme is called by a cytokine, and the cytokine is made by the Speck. If that chain holds, then the structure whose loss defines the transition and the organelle whose activation defines the disease are the same lesion, read from two ends.